The white oak is one of the most abundant acorn-bearing trees in Virginia. Acorns are an important facet of tree distribution and have a major impact on the food web. Due to their ecological significance, it is important for ecologists to understand what factors influence the total mass of acorns available to the ecosystem. And while there is extensive research on the number of acorns produced by white oaks under various circumstances, the size of the acorns has not been investigated. One factor that may correlate with acorn size is the size of a parent tree. Tree diameter can be an important factor in determining tree health and age. This can be affected by genetics, environmental nutrients, water, climate, and even events such as forest fires or logging. To understand the role this may have on further tree distribution, along with current scavenging animals, we must assess whether parental tree diameter correlates with acorn size. We hypothesized that larger trees would produce larger acorns based on the assumption that larger trees have greater amounts of metabolic resources. To test this, we measured the size of acorns and compared them across parent trees. We selected 12 white oaks of varying sizes. The trees are selected at one of 3 locations that are all standardized to reflect white oak's ideal environment. Kew Alba typically thrives in well-draining soil, so each location consisted of well-draining slopes within the 1,600 to 1,800 foot elevation range. At each tree, size and location data is recorded and 3 acorns are collected for further measurement at the lab. The variables quantifying the tree's size are height and diameter at breast height. Acorn size is measured by length, width, and the mass of each acorn after dehydration. At each tree, after elevation and coordinates are recorded from a GPS, the diameter is measured at breast height using a DBH tape. Next, the height is calculated using a tangent height gauge and a tape measure. In addition to the experimental variables, the trees are also scored to assess age. This is expected to correlate with diameter and should be checked to understand the overall health of the tree. The hand corer is drilled into a tree to remove a sample. The sample is then stained and the rings are counted under a microscope to determine age.
The acorns were measured using a caliper to assess width and length and an electric balance to assess weight before and after the freeze-drying process. The freeze dryer was used to extract water from our acorns to get an accurate measure of weight. During the analysis of our data, we found some surprising correlations and some that were expected. As expected, as the tree age increased, tree height and DBH also increased, which shows the typical positive correlation between tree growth and size of the tree.
To ensure the acorn size is not determined by tree age, length and width values are plotted against parental tree age. There is no significant correlation, especially in trees older than 50 years. This means variance in acorn size is related to another factor.
When looking at the DBH and height versus biomass, the biomass seems to get lower as the DBH and height increases, demonstrated by DBH of 68 centimeters and an average biomass of 0.7 grams, and DBH of 55.3 centimeters and an average biomass of 1.43 grams. To confirm or deny this finding, more research will need to be done. The hypothesis is also not supported by the data for length and width. Tree height has almost no correlation with length or width, in fact. More data points are needed to establish whether the slight correlation has any statistical significance. Tree DBH reflected the same pattern, but acorn length had a slightly stronger negative correlation. Overall, the data collected does not appear to support our hypothesis. To further examine the findings of this research, more data will need to be collected. To get a more accurate understanding of tree size and its correlation to acorn size, more trees should be selected and more acorns should be tested. Other variables that should be tested include soil pH, soil moisture content, soil nutrient content, tree foliage cover, and scavenger activity. A large foliage cover allows for the tree to drop acorns far away from the parent tree, which allows for a larger population distribution. However, a large foliage would also take time and energy to grow, which can be a drawback. Acorns are a valuable food source for rodents, birds, large herbivores such as deer, and insects. The acorns are a critical part of the food web in a forest because they feed the primary consumers, which in turn feed the secondary consumers and apex predators. With a severe lack of acorns, a forest's food web can be thrown off. A food web that is unbalanced can lead to chaos in population dynamics. Acorn size plays an important role in oak tree offspring survival. Knowing how acorn size is impacted by tree size can give scientists insight into how oak trees grow and how populations of oak trees can be distributed. While having a large acorn is beneficial to the chance of offspring survival, the downside to a large acorn is the amount of time and energy needed to produce a larger acorn. A larger tree can only produce so many acorns with the energy allotted for acorn production. The larger acorn is better for offspring survival and chance of spreading the genes of that tree, but it is a costly endeavor.